Good morning and welcome to today's MPG Primer. We are very excited to be, for one of the first times this year, having a combination of an in-person talk and people listening from elsewhere. So thank you for joining us in whatever format you happen to be joining us today. We're also extremely fortunate to have our speaker today, Anika Gupta, who is a bioinformatics PhD candidate at Harvard, who's working at the intersection of computer science, statistics, and single-cell genomics. She studied computer science and biology as an undergraduate at MIT. Anika previously also served on the steering committee for the Models, Inference, and Algorithms seminars here at Broad, and she is a long-distance runner, which I was excited to read as well. So thank you so much for being here today, Anika, and we're really excited to hear about your work on single-cell data. So thanks very much. And I also wanted to remind the audience, as we always try to do in primers, that we welcome questions rather at any point. So if you're here in the room and a question occurs to you, please just raise your hand and we will make sure you get the microphone. And if you are listening from elsewhere, please just post in the Q&A, which Sarah will be monitoring, and we'll make sure your question gets voiced here in the room. So again, thanks so much, and please get started when you'd like, Anika.
Thank you so much for that kind introduction, and good morning, everyone. It's great to be here, great to be presenting at MPG. As Dan was mentioning, I'm a PhD student now in my 5th and final year in the Bioinformatics and Integrative Genomics program. I am co-advised by Eric Lander and Shoma Ray-Chaudhary, the latter of whom will also be speaking at today's main session. And in today's talk, I'll be talking about actually the two main projects I've been working on throughout my PhD, which are linked with this overarching goal of trying to systematically infer what I call directional effects from single-cell data. So this is relatively high level in its description, but we can dive in. And the context for this is that, as many of you might be familiar with, single-cell sequencing has been incredibly powerful, has been increasingly ubiquitous in its use and scale and ability to answer questions over the past few years. However, sometimes what we're left with from single-cell studies is mere association. And what if we could actually leverage single-cell data, supplement it with additional information, and get more at the direction of effects or even causality? So let's dive in. The context for this work is largely driven by autoimmunity. So autoimmunity is a phenomenon that you might be familiar with, which can lead to a variety of debilitating conditions. These can be localized, such as rheumatoid arthritis, which can lead to inflammation in the joints, or more systemic, such as lupus. And of note, a lot of these indications remain with high unmet needs. So there are therapies to address a couple of these, but they're quite, the quality of life is impacted severely for a lot of these indications, and there still aren't cures. So how do we get a better understanding of the molecular underpinnings of these diseases? Well, to start off with a little bit more at the molecular level, autoimmunity is a result of an imbalance in immune activation. So in healthy cells, we have a stimulus, which could be a foreign antigen or an aberrant cell. That is identified by an antigen-presenting cell, which then presents that to a naive T cell. In turn, this T cell releases cytokines and can differentiate into an effector T cell. An example of an effector cell is a T peripheral helper cell, which will become relevant later in our story. Now, in healthy cells, we also have a population of regulatory T cells whose primary purpose serves to create some balance between the level of immune activation and the level of immune sort of suppression. This is all great in healthy cells. However, in autoimmunity, this balance is lost. So if you look between the effector and the regulatory T cells, it could be the case that either in the absence of a stimulus, we have fewer Tregs, the fewer suppressive cells, and more or excessive effector cells, or we could just have an aberrant cell function for one of these two cell types. In either case, some of the balance is lost, and it's up to us to try to figure out exactly what's causing this. So here's where single-cell studies come in. Single-cell data have identified some immune cell types that are either expanded or depleted in autoimmunity. So here we have an example from this past year, which was a case study in lupus of over 1 million immune cells that was able to identify certain populations that are either expanded or greater in abundance in those individuals with lupus compared to those that are controlled. So the couple of examples we have on the top left We have, for example, CD4 naive T cells, which were shown to actually be depleted in individuals with lupus compared to healthy controls. And then we also have some phenotypes here of cytotoxic and exhausted phenotypic T cells that were shown to be expanded in individuals with lupus. So this is incredibly powerful and starts to pinpoint what populations might be playing a role in the inflammation. However, distinguishing causal cell states for inflammation from cell states that show up as a result or as a consequence remains challenging if we're only relying on functional data. So this is where another tool can come in. And this tool would be genetics broadly. Many of you might be familiar with genome-wide association studies or GWAS. And the key idea here is that people have been looking now for decades at loci across the genome. And for a given trait, you can ask the question, is a given SNP or a given locus significantly associated with that trait's phenotype? You can look across large populations of individuals. We now have massive cohorts, from example, the UK Biobank, which has just around 500,000 individuals. And we can ask the question, which SNPs are actually rising to the top with respect to their genome-wide significance? So on the right, you see this plot that shows a cutoff that shows certain SNPs are significant at the genome-wide scale, certain others are not, but they all might hover near each other. Many current studies focus on genome-wide significant SNPs and their effects on individual genes. And this has been incredibly useful and has pinpointed really clean stories, I think, as to what mechanisms might be at play. However, there are also many small effect SNPs that might still contribute broadly to, for example, the genetics of heritability. And this might happen more on a global scale rather than through a single gene. So to give a couple tangible examples of this, back in 2013, there's a study done using chromatin marks in different tissues that basically showed that if we look for the enrichment of disease-associated SNPs in specific tissues, we can find associations between a particular tissue and a particular disease that basically claim that that tissue might actually be important for that disease. Here we have two examples, the top one in green, bottom in orange. The top one shows that the adult liver, it was a tissue that came out from the study as enriched for SNPs associated with LDL. On the bottom, and in the context of autoimmunity, was an example of regulatory T cells that were shown to be enriched for SNPs associated with rheumatoid arthritis. So this is great. It's a really powerful example of using chromatin information to identify at the bulk tissue level associations with diseases. Now, going one step further, there's another study done a little bit more recently in 2018 by Hilary Finucane et al. That looked at SNPs that were associated either nominally or genome-wide and asked the question of which tissues were enriched for disease-related SNP. And here they were able to find, this is a subset of examples where they found, for example, the CNS tissues were enriched for SNPs related to schizophrenia. Blood or immune cell types were enriched for SNPs related to rheumatoid arthritis, musculoskeletal for height, so on and so forth. And so all of these, if you think about it, make intuitive sense, but it was really useful to actually see that the genetics recapitulate these tissue and disease associations. And even more recently, this past year from Malka's Price's group, was work published that used the tool stratified LD score regression, which I'll talk about in a bit, to identify autoimmune disease critical cell types. And they looked at a couple of different types of complex traits. As an example here, they found, say for ulcerative colitis and IBD, B cells tend to be enriched for heritability compared to for eczema and for celiac disease, T cells were enriched for heritability. And so here people, as you can see, are starting to hone in on the more granular, nuanced populations of cells that might be important for a given disease. But what if we live in a world where it's not just that T cells are important for RA or B cells are important for ulcerative colitis? In fact, if you think about targeting the entire population of T cells, you're likely going to have lots of toxic effects and side effects because there are some populations of T cells that you want to maintain their function. And so these learnings have largely been limited to either tissue or cell type level inferences. But what if we could actually identify the cell states that are enriched for disease heritability? So here again, we have the genome-wide association examples and a toy example cell type here, where within that cell type, imagine that there's a continuum of cell states. And we can actually ask the question, are there certain populations of cells that are enriched for these small effect SNPs and others that are depleted for this. And the key idea here is that a lot of these SNPs might contribute to heritability, not through a single gene, but through these more global cell states, which could then be therapeutically targeted. So this is effectively our goal. It does remain challenging, and some of the key challenges are that 90% of disease heritability lies in non-coding regions of the genome. And so approaches that use only transcriptional information are likely to miss some of these important regions. Further, chromatin accessibility profiles that might be important for disease tend to be both continuous, so not discrete in nature, and also shared across different cell states. So the work I'll talk about today in the first part is proposing a framework that integrates multimodal single cell data and disease-associated loci to identify the specific cell states that are enriched for the genetic susceptibility of autoimmunity. The first step in this process is modeling what we call dynamic regulatory elements. And by this, I mean cell state dependent chromatin peaks. And so here we have an example of a couple of cell types. The data type that we're using is single nucleus multimodal sequencing. So for every given cell in our data set, we have information for both the RNA and the ATAC level data. The RNA information is useful to define within a cell type the fine-grained axes of variation. So how are the transcriptional space changing, and what does that landscape look like? And then the ATAC information is helpful because it captures all of these non-coding regions without requiring any gene-to-peak linking assumptions, which have been quite popular, but require assumptions in the process. So just to expand on that a little bit, within each cell type, we can use the RNA information to define the top axes of variation. And in this case, it's simply PC. So the top principal components that describe the variation. And then on top of that, for every peak that is accessible or open in that cell type, we can track its accessibility and how that changes along these axes. So we have two examples here of what we call an invariant peak or a dynamic peak, depending on whether or not their accessibility changes with the cell state. And so the dynamic peak is increasing accessibility from bottom left to top right, while the invariant peak stays about the same. And then finally, how do we actually connect the single cell information with genetics and with heritability? We can then annotate the genome based on sets of peaks of interest. So we say we can take all dynamic peaks or all invariant peaks and ask the question, when we partition heritability, what is the per SNP effect size of this annotation, or how much is heritability enriched in this set of peaks? So we can use this framework, and just to make it a little bit more tangible as well, the exact data that we're using in this work comes from the synovial biopsies of 12 donors, 11 of whom have rheumatoid arthritis, 1 who has osteoarthritis. And so this is actually really critical because the data come from inflamed tissues rather than from blood. And so the presence and expansion of cell states that might be important is greater in these tissues. And this results in just under 30,000 cells across 5 different cell types that we looked at. The cell type labels were determined by my lab mate and collaborator, Katie Wynand, and we used the AMP consortium's larger reference data set in order to have high confidence mapping. And so if we now look within every cell type, we can ask the question, for every peak that's open, how does its accessibility change according to the top 10 PCs in the transcriptional space? And for this, I used a Poisson generalized linear model framework. Happy to expand on that further. Feel free to ask questions. Yes.
Dynamic versus invariant peaks were defined.
Yes, exactly. So that's actually really related to this regression. And so dynamic and invariant is fully in our context, in the context of the cell states that we've defined. So we've defined the top 10 transcriptional PCs, and each peak will either change in its accessibility related to those PCs, or it will not. And the yes or no answer to that question comes from this regression. So on the left, we have an example where we have the PC loading for a given PC. And then we have on the y-axis, we have the peak accessibility. So say we're looking, maybe like we go from bottom to top in these T cells as a simplification of this. If you see that a peak is actually increasing its accessibility within this, then we would call it and it's statistically significant with this regression, then we would say that it's dynamic or cell state dependent. And the other thing to note here is that we do also account for technical effects, such as the total peak counts in the donor, which could be confounders. So using these two definitions of dynamic and invariant peaks, again, these are relying on how we define our cell states. We can then look at a couple of examples. Here we have one example of the top principal component in the RNA space for T cells. And on the left, we have a UMAP of T cells colored by their embedding or by the loading for PC1. On the right, we have two examples of peaks, one of which was shown to be associated with PC1, so it was a dynamic peak, and one of which was a not dynamic peak or an invariant peak. And as you can see, the accessibility of these peaks sort of tracks based on whether or not they're associated with PC1. And interestingly, the peak that is dynamic happens to fall in the promoter of the gene CCR6, which is involved in T cell migration. In contrast, the peak that's invariant falls in the promoter of a gene that's involved in splicing. So the next step, once we've now identified our dynamic and invariant peaks, is to then annotate the genome based on these peak sets. So here, say we have the genome that has a set of SNPs for a given trait that contribute to its heritability. This is unknown to us a priori when we're making our annotation, but the idea is that we can identify the peak sets and the regions of the genome represented by those peaks. We can then annotate every region that corresponds to a peak in that set with a 1, and every other region is 0. So just a simple binary annotation. Step is we would like to partition heritability across the genome based on this annotation. So we would essentially like to identify the per SNP contribution to disease heritability. But a key challenge here is that how much causal variation a SNP tag is strictly proportional to the LD scores. So what I mean by this is that SNPs that are in linkage disequilibrium blocks or that tend to be really close to other SNPs are more likely to tag causal SNPs simply due to their proximity. In contrast, we don't actually want to be calling a SNP as having a high effect just because it's close to a causal SNP. And so we would like to stratify and account for this LD. And so here we have an example of SNPs that are either in LD blocks or lonely SNPs, is what Hillary called it initially. And you can see that the probability of a SNP in an LD block with a causal SNP is much higher than those that are lonely. So how do we account for this? In this work, I've been using a really popular tool called stratified LD score regression, which does exactly what it sounds like, which stratifies on the LD score of a particular SNP in order to identify the causal contribution of SNP genome-wise to a trait's heritability. So here you can see that they effectively regress the chi-square value on the level of LD for a given SNP. And that gives you a sense of the causal effect. So let's look at some results. Here's an example of now a dynamic and invariant peak in T cells. And if we were to ask the question, let's compare dynamic versus invariant peaks, starting off with T cells, but now looking across 12 different autoimmune and allergic diseases. Is there some disproportionate explanation of heritability between one of these annotations over the other? And this was one of the first results we saw, which is that strikingly, there is this big difference between what we see in purple and gray, where purple represents the dynamic peaks and gray represents the invariant peaks. So this suggests that most of the heritability for these 12 conditions is present and captured by the dynamic peaks. Now, if we expand this beyond just T cells and we look at all of the 5 cell types that I mentioned, we see that there are 2 main cell types that stand out, T cells included and also myeloid cells. So here the identity line would indicate that dynamic and invariant peaks capture about the same amount of heritability. And any points that are above the line indicate that dynamic peaks are capturing more heritability. And so what we can see is for T and myeloid cells, for most of these 12 traits, dynamic peaks tend to capture disproportionately more heritability. So this hints at the fact that it may not just be that having more T cells or having more myeloid cells is what is important for disease, but rather there are certain subpopulations within these cell types that may be capturing more of the signal. So the next step was to try to actually represent these populations. And ask if we were to overlay these dynamic peaks onto known discrete cell states, how can we compare the heritability that's present in each? This brought us to the next step. And again, this is in collaboration with my lab mate, Katie, who was involved in identifying the cell states in a discrete manner within each cell type. So here we have T cell states, which are represented with their most similar dynamic peaks. Katie used a label transfer approach with a large size peak reference data set to identify what cell state each cell belonged to. And then on top of those cell states, I basically identified the top 25% of dynamic peaks based on their accessibility. So as an example, the dynamic peaks that were used in the annotation to represent these purple cells, which were both T regulatory cell populations, tended to have greater accessibility in these cells compared to all of the other cells. And so with this, we could then ask the question, which of these cell states is enriched for heritability? Is it sort of similar across all of the cell states in T cells, or is it really unique to certain populations? And we started off with rheumatoid arthritis. And what we found was that there are a subset of cell states that tend to have high per SNP effect sizes, high and significant. And the rest of these cell states actually don't seem to harbor heritability signal for rheumatoid arthritis. And if you look at the highlighted cell states here, they capture two main populations, which may look familiar given the earlier slide, where I talked about the T peripheral helper cell and its cousin or a related cell state, the follicular helper cell, both of which are effector T cells. And then also in purple, we have two T regulatory cell populations. And so if you might remember, they play sort of complementary roles with respect to activating and suppressing immune function. And so those are the cell states that are popping up as having the highest, most significant effect sizes here. But if we were to expand now to all 12 of the traits that we look at, we also see similar results. We see that, in fact, the same effector populations and the regulatory populations seem to have the most heritability signal. So this can start pinpointing what are the actual cell states that might be involved in the genetics of heritability and could thus be therapeutically targeted.
What would be the impact if you used a proportion different than 25% to define your most variable? Did you see an impact of that choice?
Yeah, that's a great question. Yeah, so I also looked at smaller annotations. And so I looked at 10% most similar. So that made this, this overlap in the potential similar peaks much smaller and made the specificity much greater. And what we saw was that the effect sizes, for example, the overall effect sizes tended to be slightly lower, but the overlap and the uniqueness between them. So say if you were to condition on the signal for one cell state and looked at the residual signal in another, that tended to be greater. And so we see the similar trend that we saw in rheumatoid arthritis across these 12 conditions. And so we also wanted to look at this for myeloid cells, which is the other cell type that had disproportionate information in dynamic peaks. And once again, these peaks can then point to discrete cell states that might mediate the autoimmune disease heritability. So here, what it pointed out was two broad populations, one of which is the STAT1 CXCL10 positive population, which is very similar to a population found by a member, an alum of our lab in this past year that was shown to be expanded in a couple of autoimmune conditions. And now we actually have heritability information to link it to the disease as well. And then we also have the tail of these cell states here are entirely dendritic cell populations. And so there seems to also be heritability signal in these populations. And finally, we started to explore, now that we've identified certain cell states that might be of importance, can we start to pick at what are the actual mechanisms, what are the programs that might be involved in linking the SNPs to these cell states? And so here what we have is on the top and bottom, we have two examples of different cell states within myeloid cells. And we have in the second column, two genes that have actually both been associated in previous GWAS with the autoimmune diseases shown on the right. And on the very right column, we have two peaks whose promoters who overlap the promoters of each of these genes and who also fall in the top 10% of similarity for for the cell states that are shown over here. And so this kind of association can start getting us to a little bit more mechanistic insights as to, okay, the cell state is enriched for heritability. What might be the genes that play in that cell state? What might be the peaks that might be playing a regulatory role, so on and so forth. But key takeaways on this part of the project, which involved estimating the cell state heritability from this multimodal single nucleus data, include that dynamic peaks that are defined along the RNA axis of variation can actually implicate which of the discrete cell states harbor the most heritability. And then these cell states might play a causal role in autoimmunity and could be therapeutically targeted without having to focus or identify a specific gene. And then lastly, while I've been talking about autoimmunity and rheumatoid arthritis, this approach can theoretically be expanded to any other complex trait. It just requires GWAS summary statistics and this access to multimodal data. So as a key paradigm, the associations I'm talking about are SNPs to cell states to them leading to this complex trait phenotype. And how can we better understand and bridge these links in a more confident manner? Okay, so transitioning a little bit, let's say we have identified a cell state of interest. So everything I've been talking about so far is in the context of a disease. How can we narrow in on not just the cell types or the tissues of interest, but a specific cell state? Say we've identified that cell state, and we now would like to identify what are the active gene regulatory programs within one state. So instead of looking across different heterogeneous cell states, we actually want to focus in on just one cell state. So the question here is there a direct regulation between a given pair of genes? And if so, in which direction does it exist? So we have a toy transcription factor and a potential target gene here, where we would like to find out if this transcription factor truly regulates that target, or is there reverse regulation, or is there no regulation at all? And we would like to do this in a systematic manner for all of the pairs of genes in a population of cells, but at steady state. So the work I'll be talking about now references work that was published this past year. And the two key ideas here are that we can leverage biological stochasticity or randomness, which I'll expand on, and time as well. And so this is, yes, from our paper from last year from, work for the past few years that has now been released. So feel free to take a look and dive in a little further. So the context again for this work is we would like to systematically infer TF and target relationships from single cell RNA sequencing data. So for cells at steady state, can we systematically learn these regulations? There are a couple of challenges for this. The first of which is that in a population of cells at steady state, there's no global or dynamic structure. A lot of experiments will rely on knocking out a gene to see big changes or studying a dynamic process such as development or differentiation, which can be hugely helpful to look for changes. For cells at steady state, we're missing all of that. Finally, although I'm talking about a single cell state, for those of you who worked with single cell data, you might be familiar with the fact that it's hard to necessarily isolate one particular cell state. And so you still have heterogeneity, and you might even have some cryptic cell states present. Here we have a classic example of Simpson's paradox, where you have on the left the expression of two different genes, gene X and gene Y. On the right, if you were to plot a scatter plot of their expression across cells, it would seem that maybe they're positively correlated, but in fact, if you overlay the two different cell state identities, it looks like, in fact, the association you're seeing is a result of having multiple cell states rather than there being regulation. So this is, again, another confounder to watch out for. Let's focus on the tools that we can use. The first is what I called stochasticity. And this is leveraging a concept known as transcriptional bursting. Transcriptional bursting is a concept that's been shown since the 1990s, mid-1990s in bacteria, to be a stochastic rather than a regular or a periodic process. So here we have an example from the top row that shows if transcription was non-bursting or regular in nature, you would expect that over time, RNA for a gene would regularly be produced, transcripts would be synthesized at a relatively periodic intervals. In contrast, the bursting model says that transcription for a gene switches between an on and off state, such that when the transcription is off, no transcripts are being produced. But then with some probability, it switches onto this on state, and suddenly many transcripts are produced in bursts. And this is actually critical because bursting allows for heterogeneity even across cells at steady state. So even if cells are the same type, same state, you would still expect that some cells have more transcription and some cells have other due to the stochasticity. Here's an example. Now I talked about how this was first identified in the 90s. It's now been shown all the way up to human cells. So the study was done a couple of years ago in the mammalian liver that was looking at single cell variability and modeling this process of bursting. I like this experiment because it captures sort of what a lot of the field used to use to identify bursting, and it leverages single molecule fluorescence labeling. Specifically in this case, they've looked at a single gene, PIC1, and have shown its expression across this population of cells. So as might seem clear just by the number concentration of red dots, some cells have more transcripts of PIC1 and some cells have fewer. What the authors did with this then was they plotted the distribution of transcripts across the cells in the population and asked which model better fits this distribution. In red, they tried a one-state non-bursty model. So the idea is that in this case, the gene would just be bursting at some periodic regular interval, and that would have led to a very normal distribution, pretty tightly centered around 600 as you see here. However, what they found was that the two-state bursty model, where transcription switches between this on and off state, much more accurately captured the transcripts that they saw. And as you can see, this distribution is highly overdispersed, so very spread out. And again, that once again supports this idea that even for cells at steady state, there is this great variance in their transcript abundances. So walking us through a little bit more, and not just relying on bursting, but now connecting the regulation piece of this, transcriptional bursting of a TF, as I was mentioning, leads to heterogeneity in the transcript abundances across a population of cells at steady state. So here we have a single cell undergoing the central dogma for the transcription factor, where it goes from the TF mRNA, gets translated into protein, which then re-enters the nucleus, binds to the target, and the target then undergoes transcription.
Anika, I think there's a question. Please go ahead and go to the microphone.
Just to get some clarification, when you say steady state, are you talking about the cell proliferation cycle and it's at M phase or S phase? Is that what you're referring to, or what does steady state actually mean in this case?
Yeah, that's a great question. So we're talking about in the absence of any external perturbation, and that could be experimental or that could be biological. So in the absence of some change like differentiation or development, and in the absence of any perturbations that we have introduced.
So if that's the case, then how do you distinguish when the cell is actually proliferating? So it will go through various cycle, and then a cycle will come when it's not doing anything. So when it's actually generating, it's gonna do more than when it's at rest stage. How do you distinguish between that stage?
Absolutely. So that's a great question, and one that we thought about. So the cell cycle is definitely a change, or it introduces a change in cell state. And the key difference there is that the cell cycle tends to happen on the order of, say, maybe a day, or it happens on some time order of magnitude. The time that we are talking about for regulation is much shorter. And so I'll be talking about sort of the response curve that we expect to see, the time delay with which we might expect to see this response. And that tends to be shorter on the order of maybe 8 hours between a transcription factor and its target. And so the second point that I'll mention here is leveraging time, and being able to then look at multiple time points helps distinguish cell state differences between regulation.
I see, thank you. But why I was asking is because when in lab people do this work, they will change the media and all and everything. So how does that impact? And there must be some other ways to normalize that impact or take away or regress it out. That's what I was asking.
Right, no, absolutely. And so that will get to, so I mentioned the sort of presence of multiple cell states. That could be a biological cell state, that could be an additional confounder. What we would expect is that for any cell state that is broadly called cell state that is present for a longer period of time, you would expect that the patterns would look different, the covariation patterns would look different over time. So I'll talk about that, but please feel free to follow up if I don't address the question. And so the idea here is that cells that had many bursts of the transcription factor will have more transcripts for that TF, more protein molecules, and then as a result, more transcripts for its target, compared to those that had fewer bursts will have fewer of all of these things. And so, as I was mentioning, there's a sequential nature of how all of this happens related to the central dogma with translation. So the transcripts then transfer the information to the RNA, to the protein, which then goes and binds to the target and affects the target RNA. So this brings us to the second concept that we leverage, which is this idea of using time and looking over time. And the key idea here is that information flow can actually occur across variables over time. And it's a concept that's been widely used in dynamic systems. So if you're looking at like differentiation, for example, you can see that cells at state 1 or genes at state 1 can actually share, and then with some time delays, transfer the information to other genes at a subsequent time. And so here's an example of, say, gene X regulates gene Y. You can measure gene X abundance at some earlier time, and then with some delay, you can wait and measure gene Y's abundance. And if you were to look at the correlation between those two time points, you would expect that to be high. So as I mentioned, transcriptional heterogeneity is propagated from a TF to its target genes in sequential steps, each of which of these creates this time delay. So here we have a toy example of a population of cells. And if you were to split them by those where the transcription factor was bursting through this initial time point T0, and those where the transcription factor wasn't bursting, you'll see that there is a sharp increase in the number of TF transcripts that then decreases due to decay. This is then propagated to the protein, which increases in abundance due to translation of the transcripts, and then starts to decrease with decay at a longer time scale. And then finally, you have the target RNA, whose transcripts increase with some delay that sort of follows the protein abundance and then decays as well. And so this curve is the key shape that I'll be talking about. And this starts to point to what the gentleman asked here, where if you look at the transcription factors RNA abundance at time 0, and look at the target or putative targets transcriptional abundance at time 2, you should expect to see, with some time delay, some strong association. And so say we take the two snapshots here of the TF at the earlier time point and the target at the later time point, and you would just look at the scatter plot of their abundances with this time delay, you would expect to see high correlation. And interestingly about this curve, it increases sort of in relation to the abundances and the propagation of these steps, and then it decreases over time. And I'll talk about some of the factors that influence that. So the other key component that addresses this question as well is this concept of distinguishing between cell state and direct regulation. I adapted this from work that was published now over a decade ago. They were ahead of their time and ahead of the technologies that existed. And it was all theoretical, but the key ideas here were that if you have direct regulation and you look at the correlation between the protein levels of two genes and their mRNA levels, you would expect it to be pretty high for the protein and a little bit noisier for the RNA because there's a step in the middle that you're not capturing. If we were to look over time now, you should expect to see that there's a similar shape in the abundances, but it's delayed and it's stronger for the proteins than for the RNA. Now, if you have cell state-based covariation, so anything that's more globally affecting both X and Y, then you would expect that measuring them at the same time would have a really strong correlation. But then if you look over time, you would expect that their patterns and their trends would completely overlap each other because they're both being affected by the same external factor. And so our solution to distinguish covariation that is related to the presence of multiple cell states versus regulation is to look again at the shape of this curve. And so in purple is what I talked about before. In the two gray versions and the two gray lines, the top one indicates that there is the presence of multiple cell states. So if there's this external force, you would expect that the correlation between gene X and Y would be high and it would stay high as long as that state is present and affecting both. And then the line at the bottom indicates that there's no regulation, and we're also just within a single cell state. So how did we actually model this? We wanted to mimic as closely as we could the key components that are happening within a cell that are involved in both bursting and regulation. So what you see here is a model with the three key entities, the TF mRNA, the TF protein, and the target mRNA. And for both the TF and target, their transcription happens again in a bursty manner. And so transcription will either turn on with some probability or turn off. When it's on, then of course transcription happens, creates RNA, some of which decays, and then for some of which translates into protein. For the transcription factor, this protein will then bind to the target. We model this as a regulatory link function that affects the target's bursting frequency, so how often it's bursting based on how abundant the TF protein is. And with that, we could then we wanted to ground this in real biological parameters. And so we looked at a bunch of experiments and found that for some of the key parameters that are involved in our model, there are certain ranges that people had found to be the most common and the most widely found. So a couple examples of these parameters are the time between bursts. So it tends to be quite long. It's on the order of hours compared to how long a burst lasts, which tends to be on the order of minutes and is much shorter. Also, things like the mRNA half-life, protein half-life will affect when and how much the peak curve or the curve peaks. So fitting this all in, we can use this model to then create simulations that result in distribution. So here what you're seeing is the steady state distributions of each of the three key entities, TF-RNA, TF-protein, and the target RNA. And this is great, but from a single time point, we actually can't distinguish correlation from causation. But if we look over time, we can start to look at the compounded effects of bursting. So here what you're seeing is following a single cell over time. The first row asks, did the TF burst at this time point or did it not? We see that when a TF bursts, the TF RNA transcripts also starkly increase before decaying. This then leads to the TF protein increasing in abundance before also decaying. And then finally, the target transcription starts bursting more. And so the bursting frequency goes up, you see more target RNA, which then also decreases. Now, if we extend this to a population of single cells in steady state, we see that this is similar to the schematic, but now in simulated data. If you're to separate cells based on those where the transcription factor was bursting versus not at the first time point, you'll see the sharp increase to a decrease, these similar trends. And the key idea here is that the information is flowing from the TF-RNA to the protein to the target RNA. And one thing to note here is that you can track both. Depending on the kind of single cell data you have, you can track both the total RNA abundance and the nascent abundance, which gives a more localized, acute picture of the regulation that's happening. So as I mentioned, this information flow does undergo a time delay. And so looking at multiple time points is critical to be able to distinguish what the shape of the curve is and distinguish regulation from cell states. So each of these steps happens at a slightly different time point. One of the key things to note, again, we have this purple curve, which indicates, we're looking at the TF and protein relationship, but it very closely mimics the TF and target relationship. And what you'll notice is that it starts to fall off right when the TF RNA half-life and protein start to precipitously drop. So when this curve peaks and when it starts falling off is directly related to how fast the TF RNA decays, and then a little bit less related to, but still affected by how fast the protein decays. So this was all in simulations, and in an ideal world, in a clean, perfect world, but as you know, there are more considerations to be taken in experiments. And so we wanted to think through a couple on the both biological side and the experimental side, and the technical side as well. On the biological side, I mentioned in our model, we have a variety of biological parameters, some of which might affect the timing and the peak of when this curve happens. So in purple, for all of these plots, we have the median values for every single one of these parameters to show when we would expect the response curve to peak and at what magnitude approximately. What you can see, though, is that for certain parameters, such as the protein half-life of the TF, and then the burst frequency of the TF, with a longer either half-life or a higher burst frequency, it seems that the magnitude of the peak is actually much lower. And so the signal and the difference you would expect to see would be much lower. And also, if you look at a couple of other parameters, such as the RNA half-life or the target burst frequency, you see that as we have a longer half-life or a shorter or lower burst frequency, you see that the time at which the peak happens gets delayed. And so all of these parameters are important to play with to get a sense of when you might want to sample and what magnitude and how many cells you might need in order to catch the actual magnitude of difference. This is on the biological side. The other considerations are technical. So while single cell technologies have been increasingly powerful in both scale and read depth, there are still some limitations. We can't sequence infinite cells. And the other challenge here is that we would like to look at the same number of cells across time points. And so there are two key criteria to keep in mind here. One is the UMI detection efficiency. So as we can detect fewer and fewer transcripts and we have more zeros in our cells, the ability to capture the actual effect size and the response becomes lower. And so the magnitude of this response curve will go lower. In contrast, the number of cells also makes a difference, but in a different way. As you decrease the number of cells present in your sample that you're sampling at each time point, the noise of the estimates becomes much greater. And so the ability to actually see this curve nice and cleanly goes down dramatically. So key takeaways, everything I've shown here is mostly theoretical, and we've tried to incorporate some of the real considerations to implement this experimentally. Some of the key takeaways from these learnings include the fact that the stochasticity of transcription leads to this heterogeneity in RNA levels of genes across cells. We can then leverage these distributions and also leverage time and look at the time delay to infer gene regulation and to be able to distinguish that from the effects of cell state. Finally, of course, as I was mentioning, both biological and technical considerations must be taken into account for experimental design that aims to infer this direct gene regulation. And my lab mate and collaborator, Jorge, has worked on a pilot experiment that's also present in our paper if you'd like to take a look. But to step back, I started off the story talking about autoimmunity and complex diseases and talking about the power of single cell data, especially nowadays in the context of supplementing it with additional information. In the first case, I talked about heritability and the fact that we can aggregate SNPs with small effect associations and use that to pinpoint cell states at a more fine-grained level that can then identify cell states that might be causing these diseases. The second project talked about once we've identified a cell state of interest, we can leverage time and natural fluctuations to help us systematically infer the gene regulatory programs that are at play. And we hope that these approaches will become increasingly useful across a variety of traits, across a variety of cell types, to look in both health and disease as both the scale and scope of single cell studies and genome-wide association studies increase. And with that, I'd like to thank both of my PhD advisors, Eric Lander and Shoma Ray Chowdhury, as well as many people in both of the labs who I've been really fortunate to work with. It's been a lot of different intellectual, interesting conversations and a lot to learn. So thank you to everyone in both of those labs. Thank you also, last but not least, to the MPG organizing committee for inviting me, and happy to take any more questions.
Thank you so much, Anika, for that really, really amazing and thought-provoking talk. I think we probably are a little short on time, so we think maybe one question would be good if anyone has a question they'd like to raise.
Hey, I'm curious if people have been able to infer the rate for switching on and off and see how that depends on different cell states and settings. Yeah,
Absolutely. So two of the parameters that I included in that table are what we call K-on or K-off, which are the rates for switching. There was a paper in 2019 that actually used single cell data to, again, find updated distributions of these. The effects of cell state, I think we've largely been looking in the context of steady state. And so I I don't know if that would, I would imagine that maybe it would have systematic effects, but that's a great question. And yeah, there's extensive literature on what the known sort of ranges are for both of these rates. And it's interesting to note that the rate of turning on is much lower than the rate of turning off, which leads to really short bursts that are very widely spread out.